8 women will develop breast cancer in their lifetime. It's an absolutely horrific statistic. And unfortunately, the existing therapies we have a limited efficacy on patients that have a metastatic presentation, meaning that their cancer has spread to other organs. Therefore, we really need more understanding on the mechanisms of breast cancer metastasis. And my PhD is trying to uncover one of these mechanisms in the breast cancer's favourite place to spread to, the bone. The tricky thing about when breast cancer cells spread to the bone is that very often when they reach it, they enter a state of dormancy, meaning that the cancer cells fall asleep, as I've depicted via the cartoon in panel 1 of my slide. These cells, they can stay in the sleepy state for up to several decades, and when they're asleep, they don't divide as much as they usually would, which also makes lots of the cancer therapies we do have inadequate because these are developed to target the rapidly dividing cells that are typical for tumours. So this maybe doesn't sound that bad. Sleepy, non-dividing cells in the bone, why the fuss? Well, the sleepy cells can unfortunately also be woken up by some yet unidentified alarm clock-like signal from within the bone environment. And it's this alarm that then causes them to start dividing again, lots and lots, forming secondary bone cancer, much like the cartoon in panel 3. The bone marrow is a complicated environment, so in the past it's been really difficult to actually pick out what signals are causing this phenomenon. However, recently it was found that these tissue repairing stem cells that reside in the bone, the stretchy looking grey cells on my slide, produce vesicles, which are like little cellular parcels that they send to the arriving cancer cells that could be causing their sleepiness. Essentially, the cancer cells eat the vesicles, fall asleep, suddenly start dividing again, and then repeat their cancer activities. They eat, sleep, divide, repeat. And ultimately, my research is looking at these vesicles, but also trying to find out what might be causing this alarm clock to go off. And to do this, I'm looking at the difference between healthy and injured stem cells, because we know that the tissue regenerative capability of stem cells actually changes depending on how healthy the stem cells are, a change that we would see after any injury or even upon the patient's aging, and that this change in the stem cells capability might be able to actually explain the alarm clock going off. My data so far strongly suggests that the vesicles from the healthy stem cells cause sleepiness and from the injured stem cells cause awake cancer cells. I'm now actually trying to uncover what's in these two different types of vesicles that might explain this mechanism. Because if we can understand what's in the vesicles, we could either engineer a treatment that causes the cancer cells to stay sleepy forever, which would allow the patients to avoid the bone cancer, or treatment that wakes the sleepy cells up early on in disease progression, which would then allow the amazing therapies we do actually have to take effect. Thank you.